This is Jishan from JC Tuition, and this video is on some basic body fluids physiology. So there are many compartments in which fluids in our body can sort of accumulate. They can be inside the cell, so that's intracellular, or they can be outside the cell, which is extracellular, so everything outside the cell. So extracellularly, they can be in this bit sort of around the cells here, which is known as the interstitium. That's the interstitial fluid, or they can be in the plasma, right? So inside our vessels. So a human body is made up of roughly 60% of water. So in a 70 kg man, that is about 42 liters of water. All right. Now, most of our fluid is actually intracellularly. All right. So that's where 2/3 of this 42 liters will be. All right. And the extracellular fluid, so everything outside the cell, holds therefore the remaining 1/3 of fluid in the extracellular space, so ECF, extracellular fluid. And of this extracellular fluid, 3/4 is inside the interstitial fluid, and the remaining 1/4 is inside the plasma. All right. So very quickly, 60% of our water, approximately, in an adult man, is water. So in a 70 kg man, there's 42 liters of water, 2/3 of which are inside the cell, 1/3 outside the cell, and of that extracellular fluid, 1/4 is in the plasma, 3/4 is in the interstitial fluid. So our body is filled with many electrolytes, and electrolytes are substances which split into cations, such as sodium ions, for an example, and anions, such as chloride ions, when dissolved in water. So this table here shows the normal electrolyte composition in our different body compartments. So importantly, note that the extracellular fluid is high in potassium and chloride ions. So the extracellular fluid is high in sodium and chloride ions, but it is low in potassium ions. Whereas the intracellular fluid is the opposite. It is high in potassium ions and actually low in sodium and chloride ions. So osmolarity is the measure of the number of osmotically active particles in a solution measured in osmoles per liter. And this is generally around 280 to 300 in each of our body compartments. We'll talk more about what osmosis and osmolarity means in the next slide. What do I mean by osmotically active particles? So these are particles which essentially draw water towards them. So here, as you can see, when we place solutes in a solution, so particles in a solution separated by a semi-permeable membrane, so on the right here we have more solutes, as we can see, dissolved. Well, what happens is the water moves towards the area with more solutes, or these solutes can be thought as osmotically active particles. So this small experiment shows that water molecules pass through cell membranes from an area of low solute concentration to high solute concentration, and this is called osmosis. We say water has moved via osmosis. Cells have all sorts of osmotically active sources. So for instance, these large macromolecules, smaller organic molecules, and small inorganic molecules. So by everything we just learned about osmosis, this means large volumes of water must be moving into our cells via osmosis, because of the high concentration of osmotically active solutes inside. So what is stopping our cells from swelling and bursting like a balloon? Well, our animal cells are very clever. So we have pumps such as sodium, potassium, ATPase pumps, and these pump out small inorganic ions. So this maintains our solute concentration on both sides of the membrane and prevents a disastrous amount of water moving into our cell. So tonicity is the ability of an extracellular solution, so everything outside the cell, to make water move into or out of the cell by osmosis. And we have 3 types of solutions that you should basically know about. So if we have the same number of osmotically active particles outside the cell, I shouldn't do this in red, should I? Let's do it in blue, light blue. So if you had the same number of osmotically active particles outside the cell as inside the cell, what would happen? Well, there'll be no net movement of water, right? So this is what we call an isotonic solution. Let's say we have a solution with more osmotically active particles on the outside, so just absolute loads of loads of osmotically active particles on the outside, but fewer on the inside. Well, what would happen? Well, water will tend to move out of the cell by osmosis, and this will cause the cell to lose water and shrink and shrivel. This is called a hypertonic solution. On the other hand, what if we had fewer osmotically active particles on the outside compared to the inside? Well, water would move into the cell, and the cell will swell and eventually burst. This is a hypotonic solution. All right. I've added this slide so that we can consider how fluid is transported across the walls of capillaries. So let's start from the arterial end of the capillary. Well, there's hydrostatic pressure, which is the pressure exerted on the walls of the capillary by the fluid due to the pumping action of the heart. And this causes fluid to be forced out, filtrate out into the interstitium. However, some of this fluid must be absorbed. Otherwise, we'd all be really, water heavy and edematous. So at the venous end of the capillary, the hydrostatic pressure is much less, as we can see. It's only 17 millimeters per mercury compared to 35 millimeters per mercury. Why? Well, this is because some of the fluid has already been pumped out. So as we move along, there's less pressure exerted by the fluid. But here in these capillaries as well, there's something called oncotic pressure, and this is provided by large molecules, mainly proteins such as albumin. So these large proteins inside here, such as albumin, and these tend to draw water back in to the capillaries. So the net pressure out is 10 millimeters per mercury, but some fluid is drawn in because the net pressure back inwards due to the oncotic pressure is 8 millimeters per mercury. And the excess fluid that's sort of lingering around here in the interstitium is drawn up by our lymphatic system. So, as you can imagine, in conditions with low albumin, such as nephrotic syndrome of the kidney, we have a lower oncotic pressure inside the capillary, so there is more net filtrate outside of the capillaries inside the interstitium, and this causes our patients to be quite edematous. Okay, so fluid volume is very carefully maintained by homeostasis by our bodies. So in periods of low volume, such as dehydration, osmoreceptors in the hypothalamus of our brain detect this. Well, they detect the high osmolarity of our blood, because if we have less fluid volume, there's going to be a higher concentration of solutes. The solutes will concentrate more, so our blood will be hyperosmolar. And our hypothalamus detects this and triggers us to be more thirsty, so we drink more to correct this fluid balance. But another thing it does is it sends signals down to the pituitary gland to secrete antidiuretic hormone, or ADH. And ADH acts on the collecting duct of our nephron inside our kidney and makes it more permeable to water. And that causes us to reabsorb water, also correcting our fluid volume. Secondly, our kidneys can detect this low blood volume via something known as the juxtaglomerular apparatus. This apparatus can trigger renin release. Now, the renin-angiotensin-aldosterone system is a video in itself, but I just want you to know that the end goal is that it produces aldosterone to be released from the adrenal gland, this system. And aldosterone increases water reabsorption in the distal tubules of our nephron. And by water again being reabsorbed, it corrects our blood volume. So that is the basics of body fluids physiology. Please visit our website for more support. And yeah, thank you for watching. Goodbye.